Assalamu alaikum, welcome to a new video where we'll continue talking about blood groups and how do we test for the blood group of a person. So in the last video we reached this table where it summarized the 4 blood groups, the antigens found on the red blood cells in each blood group, and the antibodies found in the sera of each blood group. Now, the important question here is how can we test for or know the blood type of each person? Of course, there are special blood tests is done using ready-made antibodies. So we have antibodies that are already made in the lab, and these antibodies are anti-A, anti-B, and anti-D. We're going to talk about anti-D later on. First of all, let's introduce you to antigen D. Antigen D is a new antigen other than A and B, and it's called the rhesus antigen. It's a different antigen. This rhesus antigen is distinctive from antigens A and B. So it is not related to them. We can have antigen A and at the same time antigen D or the rhesus antigen. If a person has the rhesus antigen, he will be called rhesus positive. If he doesn't have this rhesus antigen, he will be considered rhesus negative. So let's say, for example, a person having antigen A and antigen D or the rhesus antigen on the surface of his red blood cells, what will he be? A positive. That's right. Another example, if a person has antigen A but doesn't have the rhesus antigen on his red blood cells, what will his blood group be? Of course, his blood group will be A, and since he doesn't have rhesus, he will be A negative. Another example, a person has antigen B on his red blood cells, and in addition he has the rhesus antigen. What will his blood group be? Of course, it will be B positive. Finally, if a person has antigen B on his red blood cells surface but doesn't have rhesus antigen, what will his blood group be? Think about it. Of course, it is B negative, and so on. Now, let's make an application to understand how the blood type is discovered. The blood test is done in several ways, but mainly in this way. We get a glass slide in the lab and we get the blood sample. We put 3 drops of the blood on this slide. On the first drop, we add 1 to 2 drops of the anti-A serum that we saw earlier. On the second drop, we add anti-B serum. And on the last drop, we add anti-D serum. Okay, the result, let's see this result. This is a real photo of a result of blood test. We see different aspects of the blood, the drops of blood. Notice how the first blood drop where we added anti-A, look how it looks, while the other drops where we added anti-B and anti-D respectively is different in shape. If we want to draw the result, we will see it like this. We see this special aspect here on the first drop. This aspect is called agglutination. Remember what we said about agglutination? If there is presence of an antibody compatible to the antigen, and if we have anti-A, it will bind to antigen A on the red blood cells. In this case, we call this agglutination. So what happened here is that the anti-A that we added to the blood drop found in the blood antigen A. Since it found it in the blood, it bound to it and make agglutination. If we look at anti-B, the other drop, since we don't see any agglutination, this means that the anti-B didn't find any B. And finally, in the last drop where we added anti-D, there is no agglutination, which means that anti-D did not find any D antigen. After analyzing or interpreting the result, we can say that the red blood cell of this person has on its surface antigen A only. And consequently, since he has antigen A, his blood type will be A. And since there is no rhesus, no D, he is rhesus negative, the blood type will be, or the blood group is A negative. It's a bit hard in the beginning. We're going to make another application to understand it more. Okay, another application. Also, we have a slide and we add to it 3 drops of blood. The first drop anti-A, the second drop anti-B, and the final drop anti-D. The result, the first drop has a different aspect than the other 2 drops. Now, from what we learned in the first application, in which drops do we see agglutination? Yes, of course, in drops 2 and 3. The first drop, the blood is still the same, no change, but in 2 and 3, where we added anti-B and anti-D, there are agglutination. When there is no agglutination, this means that the antibody did not find the antigen. So, in this case, if the antibody here in the first drop is looking for A, the antibody in the second drop is looking for B, and the antibody in the third drop is looking for D. In the first drop, did the antibody find the agglutinogen A? Of course not, because there is no agglutination. In the second drop, did the antibody anti-B find any agglutinogen B? Yes, because there is agglutination. In the third drop, did the antibody find antigen D? Yes, because there is agglutination. So, after this interpretation or analysis, we conclude that the red blood cells have antigen B on their surface, no antigen A, and in addition, it is rhesus positive because there is the rhesus antigen. In this case, the blood group will be B+. Another application, another case. Again, we have the test anti-A, anti-B, and anti-D. The result now, where is the agglutination here in this case? Yes, that's right. In the first and second drop and the third drop, there is no agglutination. So, if we follow the same steps of thinking, the first drop we are searching for A, the second drop for B, third drop for D. There is agglutination in the first drop, so there is antigen A. The antibody found antigen A. In the second drop, there is agglutination, so the anti-B antibody found agglutinogen B, and there is no agglutination in the last drop, so there is no agglutinogen D or the rhesus protein. In this case, look here, we have both A and B antigens on the red blood cells. And is he rhesus positive or negative? Of course, he is rhesus negative. There is no antigen D. In this case, the blood group will be AB negative. And now, with the final case, the result, of course, again, we add anti-A, anti-B, and anti-D. The result here. Now, the agglutination, where is it found? Only in the third drop, where we put anti-D. Is there any agglutinogen A? No, there is no agglutination. So the anti-A did not find any agglutinogen A. The antibody anti-B did not find any agglutinogen B. So there is no agglutinogen B, no agglutination. But the anti-D made agglutination in the third drop, which means that the antibody anti-D found some rhesus proteins. So this person has no antigens A or B on the red blood cells. There are the rhesus proteins, so we call him rhesus positive. In this case, when the person has no antigens, what his blood type will be? Of course, it will be O, and since there is rhesus, he will be O positive. So we reached the end of the second video about blood groups. I hope you understood the idea. Of course, you have to make more applications, more exercises. Thanks for watching. Make sure to stay tuned for other videos on our channel. Goodbye.